Well, thank you very much. Thanks to the organizers for the invitation. It's a pleasure to be here. As the title of my talk suggests, I'm going to talk about using star proteins as antigens to generate antibodies to G protein coupled receptors. Let me start by a quick introduction to our company. So Heptares Therapeutic was founded in 2007 with the mission of bringing a structure-based drug design to G protein coupled receptors using our proprietary technology, which we refer to as the STAR technology. As part of this technology, it also not only enables us to do effective small molecule drug discovery to GPCRs, we can actually use this technology to do biologic discovery, which is the core topic of the discussion here today. We are currently located in UK, near London. We're about 70 employees. We started as a VC-funded venture, but as of February this year, we were acquired by a Japanese pharmaceutical company named Sosei Group. We're still working independently as a research and development unit. We have a multitude of products in the pipeline, in clinical trials, in different phases of clinical trials, primarily focused on Alzheimer's disease. As a business model, we effectively use our technology in collaboration with pharma partners, both for small molecule and biologic discovery. And I'll give a quick example of one of those test cases that we've done with MorphoSys, who are an antibody company based in Germany. So in case you're not familiar with GPCRs, these G protein coupled receptors, they are a superfamily of very important drug targets. There are 7 transmembrane proteins. There are about 375 of the GPCRs, non-olfactory GPCRs, that fall in 3 major classes referred to as A, B, C, depending on their molecular architecture. Most of these have known ligands, and quite a lot of information about the role they play in physiology has been deciphered over many years of research in this field. Because of their ubiquitous role in physiology, multiple processes in normal physiology, they've been linked to many different aspects of disease pathology and etiology. You can see that in the pie chart here shown. This is a research done looking at the number of molecules targeting GPCRs in various stages of development in top 10 therapy. And you can see that, for example, 40% of molecules in discovery in CNS target GPCRs. This is followed by oncology, cardiovascular, metabolic diseases. And the commercialization of that science is that GPCRs have been, will be important sources of targets. So in 2010, for example, 6 of top 10 or 60 of top 200 best-selling drugs based on US retail sales are GPCR targets. In 2012, 19% of them hit GPCR. So some of them are actually quite revolutionary drugs. Belvic or lorcaserin is one of the first truly weight loss drugs that was developed recently, and that's a GPCR targeting drug. So important source of drug, important source for drug developers. How do they function? As the name suggests, G protein, they are G protein coupled receptors. Their primary function is to transduce external stimuli into intracellular signals. And this is done primarily by binding of the ligand to the receptor, which then results in the cell cycle, in the cell signaling transduction. The GPCR ligands can be varied. They could be light photons, as we're sitting in the room visualizing the world around us as a light photon hitting the rhodopsin in the retina that results in the activation of a signal leading to vision. Then it could be odorant peptides, short peptides, long peptides, complicated peptides, neurotransmitters such as acetylcholine, dopamine, famous ones in the brain. It could be simple amino acids, complicated hormones, lipids, or intermediates of the metabolism, lipid metabolites, or chemokines involved in the immune response. GPCR signaling is primarily mediated by a G protein, a heterotrimeric G protein. So when the receptor is occupied by the agonist, it changes the state of the receptor to the active state. that engages the alpha subunit of the G protein, which then is followed by the exchange of GDP for GTP, and that results in the dissociation of the trimer. Then the G alpha subunit results in the activation of signaling cascade downstream, depending on what kind of G protein G alpha couples to the particular receptor, and then that results in ultimately impinges on the changes in the gene transcription with various different outcomes in biological outcomes and responses, depending on the setting of the activity. In terms of GPCRs, as antibody targets, so we've done little analysis of what are the important GPCRs that can be used as antibody targets. We think that there are about 100 GPCRs that have clear rationale as antibody targets for useful biologic drug discovery. 25% of those require an activating antibody, which is probably a tougher ask, and the rest would require a blocking antibody or an antagonistic antibody. Half of these have really significant validation and high clinical value. So in the background of this information is actually, and the fact that everybody here quite clear, hopefully recognizes that the biologic market is quite a lucrative market. It begs the question: why has there only been one GPCR targeted by antibody that's been approved as a therapy? And really, the reason for this comes down to the fact that GPCRs as proteins are very difficult proteins to work with. Successful antibody generation normally requires large quantity of high-quality antigen. Now, high-quality antigen, you can define it on a very rudimentary level as something that is highly purified, is homogeneous, and is stable. So you can use it in different technologies to raise your antibody. But on all of those aspects, GPCRs pose a challenge. They are complicated. They have complicated expression and secretory pathway, so that limits their overexpression of correctly folded protein. In addition to that, inherent to the biology of GPCRs is that they continuously sample different conformations. So by nature, they are actually flexible proteins. They require to have that flexibility in order to detect different conformations. And because they are transmembrane proteins, they are hydrophobic in nature. So it's very difficult to purify them in aqueous format. So they need to be formulated in various lipids or detergents. And generally, these things cause instability, aggregation. So wild-type GPCR is generally a very difficult protein to work with. Low expression levels, heterogeneous, and unstable, prone to aggregation. So the solution that we bring to table is using stabilized receptors, which we call STARs. STAR is defined as a variant of receptor that has a few point mutations introduced in the receptor to specifically increase the thermal stability of the receptor in a particular conformation. And we typically introduce 4 to 10 point mutations to increase receptor thermal stability by 10 to 30 degrees. Now, the stability, we can measure the stability in terms of increased stability with respect to chemical denaturation. We use temperature as a variant because temperature is easy to measure and is easy to control. So stabilized receptors, once we stabilize it, we sort of bypass a lot of those problems that I described with normal wild-type GPCR. So then they can be purified in larger quantities whilst retaining their function and integrity. So therefore, they become really high-quality antigens and then increase the success rate of antibody generation. How do we produce the STARs? When we start with the conformation. So this is the key. We're stabilizing the receptor in a specific conformation. The conformation is typically driven by the pharmacology of the ligand that we use. Then we make a library of mutants, single point mutations. We establish a thermal decay assay. So this is, the most basic form of it is we form receptor ligand complex. Then that's solubilized. That complex is incubated in different temperatures for a fixed period of time. And then at the end of the experiment, we measure how much ligand binding is supported by the receptor at different temperatures. And plotting that against the temperature gives you these TM decay curves, where the TM is defined at temperature at which 50% ligand binding can be supported by the receptor. So then we plug in our mutants in here. So we identify mutations that gradually right shift this curve therefore increasing the thermal stability of the receptor. Combining those mutations give us a very stable construct. In the process of doing that, we test the pharmacology to ensure that we're staying on the right path in terms of selecting the right conformation. And also we do other quality control steps, such as looking at the general biophysical, biochemical properties of these receptors using techniques such as fluorescence size exclusion chromatography. So this is an example of a receptor that's been stabilized in the antagonist conformation, and we're looking at stability of this receptor going from the wild type shown in this small curve here, and through progressive addition of the stabilizing mutations, we can see by the time we get to the final variant, the TM of the receptor has increased from 19 degrees to 41 degrees. And you can see this reported in this TM curve by this really massive increase in functional expression of the receptor and the increase in the TM of the receptor. The same information can be visualized using looking at the FSEC profile, where you can see that the receptor, now the final construct, our star number 8, can be expressed to much higher concentrations, and we have a very homogeneous sample, as shown by the sharpness of the size exclusion chromatography profile. As I mentioned, we're stabilizing the receptors in a particular conformation. In this case, this is an example of orexin 2 receptors being stabilized in the antagonist conformation. The antagonist conformation, if it's been stabilized in that conformation, you expect that receptor to be unable to be in the agonist conformation. So that's the point about conformational stabilization. And what we're showing here is, again, we're going from wild-type receptor to the final variant, and we're looking at the ability of the receptor to be activated by the peptide agonist. And as you can see, the wild-type receptor can be potently activated by the agonist peptide, but as we're increasing the number of the stabilizing mutation in the antagonist conformation, we're actually right-shifting the response curve, and in fact, by the end of the process, we've completely removed the activity out of this receptor, indicating that this receptor, in this receptor, there has been a concurrent increase in the conformational selection in thermal stability and conformational selection that can be measured by complete reduction of the receptor activity. We validated this through this technique across the GPCR superfamily. So we show, here is some of the example of the receptors we've stabilized in different conformations from different subfamilies. In each one of those, we're looking at the TM decay curve of the star in green and the corresponding wild type in red. So we have, for example, A1, a family A receptor, responds to adenosine, A2, a related receptor, again in inverse agonist, serotonin receptor in agonist, or the corresponding one in antagonist, all the way to members of the family C receptor or family B receptors, such as mGlu-R5 or GLP-1. So this method can, we can say with great degree of confidence that can be transferred across the entire family of the GPCRs. And we've made more than 50 of these different receptors in different conformations. In terms of specific utility of this star proteins for antibody generation, specifically for display technology, is that a star protein is locked in a single conformation, so therefore binders to this conformation are preferentially selected from the library that you put these in. So if you want an antagonistic antibody, an antagonist stabilized receptor is more likely to bind to those type of antibodies. So you increase your hit rate, your primary hit rate. The proteins exhibit extended durability because of their increase in their thermal stability, and then can be stored, frozen, and reused once immobilized. And you can see that in the half-life experiment shown here of one of our stabilized receptors. You can see in green, this is the star half-life, this is the wild type. Within minutes of incubating these proteins at 20 degrees, the wild-type receptor is almost completely inactivated, incapable of binding the ligand, whereas the stabilized receptor can do that in the half-life of these proteins about 18 hours at 20 degrees. So sufficient time to actually be able to do the primary screen for finding binders from using display technology. And the fact that, this is a highly purified prep means that you're actually using this, you have reduced false positive or non-specific binders. And then you have complete control over the density of this protein that you use in the in your selection screen, and therefore you can have much higher density of the pure protein compared to just expressing the receptor on the surface of the cells or in liposomes. Some of the things that we've done with the stabilized receptors for antibody discoveries that we can, as I mentioned, you can use the purified protein for display technology, or you can actually use the purified protein for in vivo immunization, or we validated this using actually genetic immunization. And then once you have the antibody specifically on the stabilized form, you can actually use this with relative ease. It's not easy, but it's much easier than wild-type receptor to actually form the antibody stabilized complex and submit that for X-ray crystallography to solve the structure of the complex and really get a full insight, detailed atomic insight into the way that your antibody and the antigen interacts. Important for further optimization of the antibody or for epitope patenting, etc. In addition to that, we use this in our small molecule discovery programs is Biocor to map the binding sites. But in addition to that, you can actually use this very nicely to define the kinetic of binding of the antibody to the protein. Now, it depends on the if, for example, if you have a situation where you want your antibody to have a really long residence time and occupy the target for a prolonged period of time because you feel that that's important to the therapeutic value of that antibody, you can optimize that using Biocor. And then, normally then what we do is that once we have, got all of those things, then we go back to the wild-type receptor, and then we test the biology of the antibody and wild-type GPCR signaling or normal GPCR binding, etc. So then, all of those things need to be reverted back to the wild-type to make sure that what we have is of relevance to the wild-type receptor. So let me just show a test case that we've done. This is a collaboration we did with MorphoSys. That they nominated a target. This was a Class A GPCR, was validated in inflammatory diseases, a range of diseases involved in inflammation, and they wanted a receptor stabilizing the antagonist conformation. And this is what we did. So this is the wild-type receptor, different variants. So the variant 4 was the one that was delivered to them. You can see as we're increasing this, with the increasing number of mutations, we're significantly increasing the stability of the receptor to variant 4. So we've gone from the TM of about 18 degrees to 44 degrees. And that's the increase of 26 degrees. The half-life is increased by about 17 hours. And I draw your attention to the Bmax. Bmax refers to the top of the curve, obviously. So this, we've gone from the counts of about 8,000 to 18,000. So that represents the increased number of functional receptor that can be expressed after a stabilization, more than twofold. So MorphoSys then used this, our stabilized receptor in various different selection strategies and have managed to raise a number of different binders, specific binders. And this is shown here. This is flow cytometry screening on the target GPCR expressed in CHO cell line using their primary hits. And this is compared with flow cytometry screen on cell line expressing a relevant GPCR. So you can see that going from 10-fold over background enrichment to 5 to 10-fold or 2 to 5-fold, they see a huge positive response when cells are expressing the receptor, but very, very little false positive response in the control cell line. So really successful outcome of selection and primary screen, very high hit rate on the cell surface expression, and very little, no detectable binding with very good background to to signal to noise ratio. And so the sequencing of this showed that they actually had identified a diverse set of antibodies. Obviously, for antibody development, you want to have cross-reactivity to cyano species. The human cyanoproteins tend to be quite well conserved, but nonetheless they need to be tested, and these were screened on a human and cyano target with an irrelevant GPCR as a control. 190 of them cross-reactive hits were selected for snapshot sequencing, and 63 unique antibody clones were selected that could detect both human and cyano. Only two of these, interestingly, had a linear epitope. The other ones are completely conformational epitope. And these antibodies are being progressed in the discovery pipeline of MorphoSys. Two of the candidates show 100% inhibition in the Discover X beta arrestin assay, and the 19 variants show more than 50%. And they are at the moment they are just progressing those various stages of their biology. So I'm going to stop here, acknowledge the people who've been involved in various different aspects of what I've discovered, as well as our collaborators, MorphoSys, and I thank you for your time. If you have any questions, I'll be happy to answer.
Okay, first of all, I'd like to announce that we do have one more speaker after we've had an update in the program here, so we will have this after questions. Do you have any questions right now for Ali? I'll start with one. I'm interested, I'm convinced from your data here that this is a very viable approach, and it's not my field at all, but it seems very exciting that we have a possibility now of more efficient application. I don't know what competing technologies exist, so I'm wondering about your business model. How will this be employed by those who are interested in such approaches?
So we work in very flexible collaboration agreements. We've done cases where we've done it's sort of a all-in collaboration. They nominate a target, we do stabilization, then they take on and do the developments, and then we have normal milestone payments or FTE covered during the life of the project with some downstream royalties, and then we have.
So I'm just curious, how would the downstream royalties work?
Oh, this is the various different, stages of if it goes first in man or first clinical.
So that would be some royalties on the discovered sequence, or
it'll be on the agents. But those are the technical details of the contract. It could vary depending on different setting in the negotiations. But we've done cases where we've gone sort of risk share basis, where we work with the partner. And we contribute to the development of these as well through the discovery and the development stage. What we have is that the technology is ours, so we own the intellectual property that comes out of this in terms of the actual core technology of Heptares.
Okay, so you would own them, the discoveries based upon this.
So the antibodies is the partner's antibody. Those would be there. So. We're doing cases where we own the antibody as well, so it depends. If you contribute to the discovery, then it would be.
Well, but if I came to you with a project and you came up with an antibody, you own it, then I could either buy it outright, or I could license it and then have payments. And if it became a therapeutic, then you would have.
Yeah, we don't have our own antibody discovery engine internally, so we do that externally. We have small molecule discovery programs. So we do chemistry, small molecule chemistry. We don't have a platform for antibody generation.
So what I see is that basically, if I come to you, virtually everything's on the table. We're going to set up the contract initially, and then live by those terms.
Yeah. Yes. Yeah.
What about my buying the technology to take home to use, or leasing it, or how does that work?
That would be a discussion to have with my business development colleagues.
Is it on the table?
You can talk to them, sure, yeah.
All right, well, thank you. Any other questions? Yes.
So, it's a very interesting system, but what type of expression system do you use?
So for expressing the receptors, we use either the insect cells or human cells. We could do both.
So equalize not related.
Sorry?
So you don't have use bacteria.
No, the problem with bacteria and GPCRs is the majority of GPCRs don't express so well in bacteria. You have to put in a lot of upfront investment in really optimizing that. So, for example, if you look at class B receptors, they have this complex architecture of the N-terminus. Those, the multiple disulfide bridges, etc., they tend to actually go into inclusion bodies, and they take the rest of the receptor with it.
It would be the chaperone to the...
You could do that, but it's very, So, but you sort of becomes, and more importantly, what you don't have in E. coli is the post-translational modifications. If a lot of our, antibody partners like the glycosylation motifs left on the receptor, so that's why we've stayed away from E. coli.
Yes.
And you showed that you can use it for X-ray crystallography. Do you already, for example, have a crystal structure of your modified star? And did you compare it to the one that was original? And are there any conformational changes?
Right. So that's our core sort of business, making on the platform side, doing crystallography. We have many GPCR structures that we've solved. We've published 2 of them in public domain. So that's we have a lot of information. So what is your question? Is it specifically?
So how are they different from the original? What exactly are those mutations that?
Right. Okay. So when we solve a structure, it's a structure of a stabilized receptors. But in cases where we've solved a structure and other groups have solved the wild type structure. Well, first of all, let me say there is no such a thing as a wild type GPCR structure out there. Everybody who does GPCR crystallography, they use some sort of a technique or a trick to stabilize those receptors one way or another. Either it's the ligand, or it's a nanobody, or it's a fusion protein, etc. So everything is done. Everybody uses some sort of a technique to facilitate.
Is it because they're responsive to the environment?
That is part of it. So the conformational flexibility is a big challenge for a structure, because that kills crystallography. So that's a part of it. And the other part of it is just a general, the other problems, just instability, etc. So a lot of groups, for example, use high affinity compounds to actually add stability to the receptor. We don't do that. We engineer in the stability into the receptor. But in those cases, we see no difference in terms of the this. Once when we solve the structure of a receptor in the antagonist conformation, it's the same conformation that other people have solved the structure of. A classic example was A2A. They solved the structure of that in the antagonist conformation, or in fact, actually active agonist conformation as well. So in terms of conformational changes, obviously with the structural biology, there is always going to be the minutiae that is going to be different from structure to structure, but the overall, the changes are as expected from conformational stabilization.
Yeah, to follow up on that, do I understand that the modifications you make to stabilize conformation will depend on which conformation you need? One conformation for the agonist, one different conformation for the antagonist?
Yes, that's true. And we actually now, since 2007, there's been sort of you might be aware of us, there's been revolution in GPCR structure biology. So we really sort of understand, especially for family A receptors, we really have a very good understanding of structural insight and rationalization of structural rationalization of different conformations. But for discovery, you get the conformations are different. Really, differences in the way that the receptor sits when it's in the agonist or antagonist conformation.
It should also mean you will be generating different type of antibodies.
Oh yes, depending on the conformation you use.
Do you have any idea what your conformation is?
Well, we drive the conformation. We know from ab initio, we know what conformation we want, and we use the ligand to drive that conformation, and we stabilize it using those. So the mutants that stabilize the agonist conformation in same receptors is a different collection to the mutations that stabilize the antagonist conformation.
Oh, good.
So really, I mean, there is a very tight link between, this idea of thermal stabilization and conformational selection of the receptors.
Does it mean that you need a crystal structure?
For what? For antibody?
For designing your mutants?
No. But in fact, actually, that's why we do the stabilization, to get a structure. We don't need that because it's an empirical process.
I mean, wild-type structure.
No. We, this is all done empirically. So we make a mutant library and we screen those for thermal stability.
How large are your libraries typically?
So we don't actually. We don't do. I talk to like antibody audiences. They have a different idea of a library. For us, a library is having 300, 400 mutants. We really want them to be small because we're not doing molecular evolution. We're actually doing single point mutations, scanning mutagenesis. And the smaller they are, the better. So now we've actually collected so much data over the years. We know, for example, what part of the receptor we can actually target to enrich for a set of mutations. We've been able to stabilize the receptor to similar extent that I showed for this target by screening 90 mutants. Thanks.